Welcome to Azad Bioacademy. Today's lecture topic is molecular genetics. Basically, today I will discuss chromosome and its associated terminology like chromatin, chromatid, centromere, histone protein, gene, DNA, like that. So, let's start. That is chromosome. So, firstly, you have to know what is chromosome and where it is located exactly. So, before giving the definition, we should know some backgrounds of it, of the chromosome. So, if you think your body has a head office, that is head. So, if you think this is head, this is head, okay. So, within the head, so this is head. Within the head, brains are there, brain, okay. And the active unit of the brain is like neuron, okay. So, if you think this is a cell, so head of the cell is nucleus, and within the nucleus, as like, so this is the nucleus, that is head of the cell, and within the nucleus, as like brain, genetic materials are there in a condensed form. So the condensed form of genetic materials is called, you can say this is the chromosome, and the active unit of the chromosome, as like a neuron of the brain, active is DNA. So you can guess DNA, the condensed form of DNA within the nucleus may form chromosome, but exactly like that. So before discussing the chromosome, let's talk about the background of chromosome. This is astonishing thing, you know, in normal state, before cell division, no chromosome is present within the cell. No structurally visible chromosome is not present within the cell. So how genetic materials basically present within the cell, and where and which form, that question may come in your mind. So that is the genetic material DNA. So the DNA deoxyribonucleic acid is the polynucleotide chain sequence, double stranded, you know the background. So this genetic material is present, and how many numbers of DNA materials are within your cell. So basically, you know, within our cell, chromosome, 46 chromosome in human eukaryotic cell, 46 chromosome are present, you know, but exactly the same is 46 DNA molecule is present within a eukaryotic cell like human cell. So 46 DNA molecule, and that each and every DNA molecule, if you think this is one DNA, this is another DNA, this is another DNA, such like DNA, the number of total DNA within the human cell is 46. So how it can be considered as a chromosome? So already I said before cell division, in normal condition, the genetic material DNA is very functioning and taking part in different expression and production of different types of protein and so on. So how it will become a chromosome and when it will become chromosome? So that is the main question. So if you think in that way, then you can get the exact chromosome things. So this is the genetic material DNA. So DNA is not present like a single DNA within the nucleus, not like that. So how DNA present? So DNA, you know, the sequence of nucleotide sequence, the DNA. So that DNA is as like a thread, as like thread. So how it will be present? So firstly, as some protein complex, a protein like histone. That histone protein has different subunit. The total number of subunit is 8, then it will form a octamer, and that octamer histone protein is one of the basic thing on which the DNA will wrap up. Okay, so like that, if you think this is DNA, okay, and that DNA is just wrapping up on the histone protein. So these are the histone protein. So on the histone protein, if you think this is the histone protein, if you just take a thread and if you wrapping off, then the thread is DNA, and this pen is the base on which the DNA is just wrapping up. So that is the histone protein, and the thread like substance is DNA. Okay, so this assembling form of histone protein and DNA, this is called chromatin. Okay, so in chromatin form, normally within your cell, chromatin form DNA is present, not chromosome. Okay, chromosome is not structurally visible during normal state. So normally the genetic material DNA is present in a form of chromatin, that is by assembling with the histone protein. Okay, because it is very easy to unwind and it is very easy to locate a particular gene that will be expressed for the production of particular protein. But when it will be more condensed, and then it is very difficult to unwind. So I will just go to the next. Okay, so this is the chromatin, and in that form normally DNA is present within the cell. So when the uh, chromosome will form, that is the question. Okay, so you know, during cell division, S phase, within the interphase, S phase, basically S phase, that is synthetic phase, over there genetic material is just copied, replicate. Okay, so if you think this is a single DNA which is just wrapping up on the histone protein, okay, and that will be replicated. That means it will be copied during S phase, and then the condensation will start. Gradually it will be visible and it will be more condensed, and then finally it will form a X-like structure chromosome. So how it will form? So go into the details. Okay, so this is a think like a DNA, and it will be copied. Okay, and then it will condense more. You know, already I covered, that is histone protein and the DNA that will form a chromatin, and then it will be more condensed and it will form a chromatid, single chromatid, like that, chromatid. And this one is centromere. Centromere. So centromere is actually The special type of DNA sequence that is responsible for capturing some uh, protein and also responsible for controlling the condensation and separation and duplication of the genetic material. That is the main point. So, the centromere is basically a particular protein, as like cohesin protein and some other protein, which is basically located within a particular specific sequence of the DNA, and this is called centromere. Okay, and the centromere, and these are the more condensed form of DNA, and that you can consider as a single chromosome, but exactly this is not chromosome. You can say this is the chromatid, and single chromatid can be considered as a chromosome. Single chromatid is considered as a single chromosome, but it will be duplicated, and the next stage it will be duplicated. If I just give you some idea, that is an S phase, that will genetic material will be copied, and then it will go toward the uh, mitosis phase, that is prophase, and the condensation will start, and then prophase to metaphase, and then anaphase, telophase, So this is a single chromatid, that is single chromosome. You can consider as a chromosome, but exactly it is not the structural X-like chromosome. So after that, what will happen? Then just go back, the structural chromatin, the twisted form, the more chromatin like will be assembling in a place like that. So if you think these are the histone protein, and the thread like uh, substances are DNA. Okay, so this is more condensed, and if you just think this is the part of like chromatid, and this is somewhat loose, and this finally is completely DNA. So this is the histone protein, this is the free DNA, you can say. This is the chromatin, the more condensed form of chromatin, that will form a chromatid. Okay, and then that chromatid will be duplicated, and then what will happen? Just follow the next. Okay, so this is the chromatid, you can say, as a single chromosome, and this chromatid will be duplicated. How it will be duplicated? Like that, and this is the centromere, and these are the chromatid, chromatid, this also chromatid, and these chromatid are identical, 100% identical, so you can say sister chromatid. This is also, you can think this is a chromosome. This is a pair of chromosome? No. This is a single chromosome. This is also a single chromosome, and this is already duplicated form, and this chromosome is ready to separate. Okay, so in generally we know the chromosome is like X structure, So this is the duplicated form of the chromosome, is like X structure. So this is also a single chromosome, as like a single chromatid, and here is the duplicated, the duplication occur. The color combination is not appropriate here. So then it will be separated. How it will be separated? So you can think like that. Okay. So this is a chromosome. This is another chromosome, separated, and then a spindle fiber will attach the centromere, and then it will be separated by the cell division. The next topic we will cover it. Okay. So the chromosome is that the very condensed form of the DNA, which you can say this is chromatid, and if you just uncover the chromatid, then you will get chromatin, and if you just pull the thread like DNA, then you will get the single DNA. So that is the single DNA, that is the assembling form of the DNA and histone protein, that is the histone protein. Okay. So this is the chromatin, and more condensation occur. Okay. More condensation occur. So this is the condensed form, and then it will form a chromatid, and then chromatid will be duplicated and it will form the X structure like chromosome. So you can say this is chromosome. So during cell division, a particular stage X structure like chromosome will form. So how many numbers of chromosome will form? So you know the 46 DNA is present within the human cell. So 46 chromosome will form like that, and then it will be separated identically into two cells. Okay. So this is the today's discussion topic, that is chromosome. It is the basic condensed form of the genetic material, and the associated things is chromatid. You know, this is chromatid, and then chromatin. Already I discussed chromatin, and the DNA, and another things you know already, the part of DNA which is particularly responsible for a particular function, or which can express for a particular protein, or which has a particular controlling ability to express any genetic materials, that is called gene. Gene, that is part of DNA which is responsible for the particular function, or you can say which can express, or you can say which is responsible for production of protein. So this is gene. So gene can be coding or non-coding gene. You know the coding gene is only 2% coding gene, that is responsible for the production of protein, and other 98% protein is non-coding gene, and that those non-coding genes, the exact function of all the non-coding sequence is completely not known yet, and some has some function of controlling or regulating some gene expression and other genetic factors. Okay. So overall, that is DNA, part of DNA is gene, and the entire DNA, entire genetic material within the organism or within the cell, that is the genome, and this is chromatin, this is the condensed form of chromatin that will form a chromatid, and the duplicated form of the chromatid, you can say this is the chromosome, and this is only visible during the cell division, and before the cell division, the genetic material 46 DNA molecule is present as like a chromatin. Okay, and these are the histone protein. So thank you very much. If you do have any question, please put your question in the comment box. So thank you very much.